In this exercise entitled Introduction to Proteomics, we are going to be analyzing the protein content of samples, particularly milk, using Bradford assay, and we are going to be confirming this using spectrophotometry. So these will be our samples, and they will be placed in containers labeled A and B. So take note of the protein content ahead of time here. The containers for our samples and our controls will be the cuvettes. So here they are, and we are going to be labeling them ahead of time. So we cannot put the labels on the cuvettes, so we're just gonna stick the label on the side and make sure not to exchange the cuvettes. So A and B will be where our milk samples will be. So here is a detail of a cuvette. This is the side. The longer side will be not the side where the light from the spectrophotometer will be going into. So this side is labeled with the triangle on top. This is where light passes through in the spectrophotometer. So our sample will be made up of a 1:50 volume ratio of the milk. So it's going to be composed of 4 microliters milk and 196 microliters phosphate buffer saline to comprise 200 microliter of the sample. So first, let's prepare sample A, or the first type of milk. So we're going to be dispensing 4 microliters of this sample into a microcentrifuge tube. So we have pre-labeled our tube with A. And again, this is milk sample A. Then we are going to dispense in the same tube now 196 microliters of phosphate buffer saline. So this is now our 200 microliter sample of milk A. Then we're going to prepare the same amount of ratio for sample B. First, 4 microliters of the milk sample B. And we have also pre-labeled this microcentrifuge tube. And then we are going to dispense 196 microliters of phosphate buffer saline with our 4 microliters of sample B. So this is now our 200 microliter sample B.